Hello friends, in this clip we shall discuss about the concept of linkage and recombination. Now, linkage is a phenomenon in which all the genes located on a chromosome are said to be linked. They are said to be linked. Now, these are the five genes A, B, C, D, E located on a chromosome. So all these genes are said to be linked and they are called one linkage group. Now here you can see a pair of homologous chromosomes and alleles of all these five genes located on them. So there are five genes A, B, C, D, E. So you'll see dominant allele and recessive alleles. Now all the genes which are located on one of the homologous chromosomes, out of two homologous chromosomes, one of the chromosomes. Okay, so all the genes which are located on one of the homologous chromosomes is called a linkage group. Now all these genes they form one linkage group. In human females there are 23 linkage groups since there are 23 pairs of homologous chromosomes. In case of males, human males, there are 22 linkage groups. Since there are 22 pairs of homologous chromosomes, this is because X and Y chromosomes in human males are not homologous. Okay, so that's all about linkage. Recombination. Recombination is nothing but the exchange of genes between homologous chromosomes. So during recombination, these homologous chromosomes, okay, they exchange their genes. Okay, it means if there is a dominant gene on this chromosome, okay, and recessive gene on this chromosome of homologous pair, then these two genes might be exchanged in such a way that this recessive allele may go to this chromosome and this dominant allele may go to the other chromosome. So recombination is nothing but the exchange of genes. It is the exchange of genes between homologous chromosomes. Now this exchange of genes or recombination occurs during prophase 1 stage of meiosis. Here you can see that dominant and recessive alleles of gene A and E. Okay, so this is before recombination, this is after recombination. So you can see the difference in the structure of these chromosomes, or you can say arrangement of genes. So arrangement of genes is slightly changed. Okay, so you can see that dominant and recessive alleles of gene A and E. Okay, dominant and recessive alleles of gene A and E are exchanged between the homologous chromosomes. During prophase 1, crossing over occurs between the homologous chromosomes. Okay, now during this crossing over, the genes between the homologous chromosomes are exchanged. One thing that you have to remember here, that the percentage of recombination is directly proportional to the distance between the genes. It is directly proportional to the distance between the genes. It means if the distance between the genes is more, then the percentage or frequency of recombination is also high. So if the distance is more, percentage of recombination is more. Okay, if the distance between two genes is less, then the percentage of recombination is less. Now, the genes on a chromosome may be tightly linked or loosely linked. If the distance between any two genes is less, you can see the distance between these two genes A and B is less, then they are said to be tightly linked. They are said to be tightly linked because the distance between the two genes is very less. Similarly, the genes B and C, they are tightly linked. Genes C and D, they are tightly linked. Genes D and E, they are tightly linked. So in short, if the distance between the two genes is less, then they are said to be tightly linked. Okay, and their frequency or percentage of recombination is very low. Means they tend to remain linked. They rarely separate. Okay, they rarely separate, or you can say the chances of their separation is very less. Right. Similarly, if the distance between any two genes is more, okay, the distance between two genes is more. Say for example, A and C, A and D, A and E, B and E. So the distance between any two genes is more, then they are said to be loosely linked. They are said to be loosely linked, and their frequency or percentage of recombination is very high. Okay. Now here, the distance between A and B is less, so they are directly linked, and their percentage of recombination is less. Okay, and the distance between A and E is more, hence they are loosely linked, and their percentage or frequency of recombination is high. A and E. Now let's see T.H. Morgan's experiments that led to the idea of linkage in fruit fly Drosophila melanogaster. He studied linkage in genes located on X chromosome in Drosophila. He conducted several crosses in Drosophila. Two such crosses are discussed here. In the first experiment, he crossed yellow-bodied and white-eyed. Okay, yellow-bodied and white. So this yellow-white means yellow body and white eye. So body color. So yellow body. So there are two body colors here. One is yellow body, another is brown body. Two eye colors: white eye and red eye. Okay. And the first experiment he crossed yellow-bodied, white-eyed. Okay, here you can see yellow-bodied, white-eyed females. So these are females because you can see there are two X chromosomes here. So this is X chromosome and this is Y chromosome. So these are males and these are females. So he crossed yellow-bodied and white-eyed females. He crossed yellow-bodied, white-eyed females with brown-bodied and red-eyed males. Now here the allele Y plus is dominant. The allele Y plus is dominant, and it controls the wild brown body colored character. Okay, so it controls the wild or brown body character, which is brown body character, which is dominant. Allele small y, okay, not small y. So allele Y plus is dominant, Y is recessive. Now this allele Y is recessive and it controls the yellow body character. Now similarly, the allele W plus is dominant and controls the wild red eye color character. And the allele W is recessive and controls white eye character. Then in F1 generation, okay, all the females were brown bodied and red eyed. All the females were brown bodied and red eyed. That is nothing but the wild dominant type. Wild dominant type. You can say all females are, okay, they show dominant phenotype. And all males are yellow bodied and white eyed. All the males are yellow-bodied, white-eyed, which is nothing but the recessive phenotype. So all males have recessive phenotype, and all females had dominant phenotype. When the F1 generation flies, they were crossed. Okay, so it means the cross between brown-bodied, red-eyed males and yellow-bodied, brown-bodied, red-eyed females and yellow-bodied and white-eyed males. Then, in the F2 generation, they observed that 98.7% of flies, 98.7% of flies were of parental type. They were of parental type. So, what was the parental phenotype? Yellow body, white eye; brown body, red eye. Now, these are the parental types. So here you can see that 98% 98% of the progeny or 98% of the flies in F2 generation, they were parental type. So either they are brown red, they are brown bodied red eyed, or yellow bodied and white eyed. So they were 98.7%. Okay, and only 1.3% were of recombinant type. That is, okay, recombinant type. So that is 1.3% were brown bodied and white eyed, or yellow bodied and red eyed. So both of them, they are non-parental combinations. These are parental combinations. So brown body, white eye, and yellow body, red eye. So they are nothing but non-parental combinations, or you can say recombinant combinations. So only 1.3% of the F2 generation, okay, they were recombinant types. They were recombinant types. Now, based on these results, he concluded that the genes Y plus and W plus, or you can say just Y and W, because they are located on the same loci. Okay. Now, he concluded that the genes Y plus and W plus were tightly linked because their percentage of recombination was very less. If the percentage of recombination is more, then they should be tightly linked. So in this experiment, the ultimate conclusion was that the genes for eye color, okay, the gene for body color and the gene for eye color, they are tightly linked. Means the distance between them is very less. Okay, moving on to next experiment. In another experiment, he crossed white-eyed and miniature winged females. So in case of Drosophila, the normal wing is dominant over miniature wing. The red color eye is dominant over white color eye. Okay. Now here he crossed white-eyed miniature winged females. White-eyed miniature winged females means they are showing recessive. Both the characters are recessive. Okay, so they are crossed with red-eyed and normal winged males. So this is the wild phenotype or dominant phenotype. So males are they are showing dominant phenotype here, and females are showing recessive phenotype. So when he crossed white-eyed miniature winged females with red-eyed and normal winged males, okay, in F in F1 generation. Okay, anyway, we shall know about these genes also. Okay, now here the allele W plus that you already know, it is dominant and controls the wild red eye color character, and allele W, okay, and the allele W is recessive and controls the, okay, white eye character. Similarly, with respect to the shape of the wings, the allele M plus is dominant and controls the wild, okay, that is normal shaped wings, whereas the allele M is recessive, okay, and it controls the miniature shape of the wings. Okay, in F1 generation, females were red-eyed and normal wing, females were red-eyed and normal wing, and males were white-eyed and miniature wing. So in F1 generation, all the females, they showed which character? Wild dominant character, whereas the all males, they show both the recessive characters with respect to color of the eye and shape of the wing. In F2 generation, they observed that 62.8% flies were of parental type. 62.8% of flies were parental type, and 37.2% were recombinant type. Okay, that is 62.8% of the flies are red-eyed and normal winged, They are red-eyed and normal winged, and white eye and miniature wings. So these are the parental phenotypes. You can see white-eyed, miniature winged, red-eyed, normal winged. These are parental. White-eyed, miniature winged, and red-eyed, normal winged. So these combinations means out of the total 100% flies, 62.8% flies they showed parental phenotypes. Parental phenotypes. Now 37.2%, okay, they showed recombinant types. That is, here you can see white-eyed normal winged, red-eyed miniature winged. White-eyed normal winged, red-eyed miniature winged. So they were recombinant type of phenotypes, not parental. Okay, they are non-parental, you can say, or recombinant types. That is 37.2%. Now, based on these results, he concluded that the genes W plus or M plus or W and M, okay, just W and M or W plus or M plus, the distance between the two genes is more compared to W plus and Y plus. Okay, right. Now, based on the results, he concluded that genes M plus and W plus, which are located on X chromosomes, were loosely linked because they were showing high percentage of recombination, that is 37.2%. So the distance between them is more compared to distance between W plus and Y plus genes. Now let's discuss about linkage maps. Based on linkage experiments conducted by T.H. Morgan, his student Alfred Sturtevant Alfred Sturtevant used, okay, he used the frequency or percentage of recombination between the gene pairs on the same chromosome as a measure of the distance between the genes. As a measure of distance between the genes. Here you can see a sample linkage map based on the results of the above already discussed experiments. Okay. Now the percentage of recombination between Y plus and W plus genes was 1.3%. Was the distance between Y plus and W plus, okay, the recombination percentage, or you can say the recombination frequency, was just 1.3%. Their percentage of frequency or their percentage is very low. Okay, the recombination percentage is very low. Okay, hence their distance is very less. Their distance is very less. If the percentage of recombination is less, it means distance between those genes is also less. Okay, so it means they are tightly linked. Y plus and W plus are tightly linked. The percentage of recombination between W plus and M plus in the second experiment, okay, the percentage of recombination between W plus and M plus genes is 37.2%. Okay, 37.2%. Their percentage of recombination is higher. So compared to this, their percentage of recombination is higher. Hence, their distance is more. Okay, that is, they are loosely linked. So that is why their distance is considered to be more. Okay, that is, they are loosely linked. So W plus, Y plus W plus, they are tightly linked, whereas W plus M plus are loosely linked. Okay, it means the gene for the color of the body, the gene for the color of the eye, they are tightly linked. The gene for the color of the eye and gene for the shape of the wings, they are loosely linked. Now here we should consider 1% of recombination as one map unit. We should consider 1% of recombination as one map unit. It is the distance between the genes. Okay, 1% of recombination is one map unit, which is also called as 1 centimorgan. So the distance between genes on the same chromosome is measured in terms of centimorgans. So 1 centimorgan is 1 map unit. That is nothing but 1% of recombination. Okay. The percentage of recombination of Y plus and W plus is 1.3%. 1.3%. Hence, the distance between them is 1.3 map units or 1.3 centimorgans. Similarly, the percentage of recombination between W plus and M plus genes is 37.2%. Hence, the distance between them is 37.2 centimorgans or 37.2 map units. Now let's solve some questions based on linkage and recombination. Now, first question, linkage group in E. coli is or are. What is the total number of linkage groups in E. coli, they asked. You know that each pair of homologous chromosomes has a linkage group. In E. coli, there is no chromosome. There is no chromosome, but there is DNA and genes are located on DNA. Since there is a single circular DNA, in case of bacteria like E. coli, all the genes are located on it, thus making one linkage group. Hence, the answer is 1. Hence, the answer is 1 because all the genes are located on the same DNA, same single circular DNA molecule. But in case of humans, what happens? The genes are located on chromosomes. Okay, chromosomes are nothing but DNA plus histones and non-histone proteins. Okay, so in case of humans, okay, and eukaryotes, okay, there are chromosomes. So on the chromosome genes are present. So if chromosomes are there, we'll consider that one of the each pair of homologous chromosomes is a linkage group. So it is not so in case of E. coli. So hence we can say the linkage group in E. coli is 1. If the question is asked like this, the linkage groups in human females 23. The linkage groups in human males 22. So like that you can answer. Now question number 2. The frequency of crossing over will be relatively more. So the frequency of crossing over is nothing but the recombination percentage. Okay. If it is relatively more, if the distance between the genes is less. No. If the distance between the genes is less, then those genes are linked, then frequency of crossing over will be less. Okay. If they are linked, the linked genes are more. If the linked genes are more, then also the frequency of crossing over will be very less. And frequency of crossing over will be relatively more if distance between the two genes is more. This is true. This is true. Okay, if the distance is more, then the frequency of recombination is more. That is nothing but frequency of crossing over is also more. Now, the map distance between two genes or alleles on a chromosome is measured in. So this we already discussed. It is measured, the distance between two genes or alleles on a chromosome, they are measured in map units or centimorgans. So your answer is centimorgan. Now, the distance between allele A and C is 4 centimorgans. Between A and B is 8 centimorgans in a chromosome. So on a chromosome, distance between A and C is 4 centimorgans and distance between A and B is 8 centimorgans. What is the sequence of these alleles on the chromosome? Now here they have given the distance between the genes. The distance between A and B is 8 centimorgans and between A and C 4 centimorgans. We know that if the distance between the genes is more, if the distance between two genes is more, then they are loosely linked. So here out of these two, you can see the distance between A and B is more. So A and B are loosely linked. Whereas the distance between A and C is 4 centimorgans compared to this, okay, A and C are tightly linked. These two are loosely linked. Okay. Now hence A and B, they are far away from each other compared to A and C. Now based on this data, we can place these genes on a chromosome. Say for example, this is a chromosome. We have to consider the distance between the genes. Okay, the genes whose distance is more. Say for example here, A and B, the distance is more compared to A and C. So A and B 8 centimorgans, A and C 4 centimorgans. So it means A and B are far away compared to A and C. So just place them here, A and B here. So between the two genes A and B, 8 centimorgans, distance is more. Okay, and between A and C, 4 centimorgans. Okay, so hence the answer is A, C, and B.